Hello, this is Dr. Clark, and today we're going to be talking about the amazing cell. We're going to start off with kind of the evolution of the cell theory, cell size limits. We're going to go into the organelles and different structures like the plasma membrane of the cell, and then go a little bit into nucleotides, and that should get us about halfway through the chapter. Of course, the learning objectives, as usual, you can read in the book, so we'll go a little bit into starting off with evolution of cells and kind of understanding that our bodies are made up of trillions of cells. We do have individual cells living in the environment around us, like in bacteria, things like protozoa. Those would be single cell organisms. But most of the organisms we end up dealing with are multicellular. They're going to be things such as dogs and cats, and us humans. We have interactions, and there's trillions and trillions of cells that make up our body. So a little bit about the evolution of cells is the first cells that we know of were actually formed probably somewhere about 3 billion years ago in the primitive oceans of Earth. And something in the environment was just right for proteins and nucleotides to be made from the carbon in this primordial soup, and cells started to develop, and 3 billion years later we're to where we're at right now. Many of these first cells were extremely primitive. They were about the size of bacteria, which is very small compared to the cells in our body, and it grew from being that small to where all at once you started having multicellular organisms, and from multicellular organisms to where we're at today in size that it's grown up. But they all started off with basic proteins, a cell membrane, and some type of nucleic acid, probably DNA, which was able to actually sit there and keep information stored on how to make proteins and how to replicate itself. So we look at prokaryotic and eukaryotic cells. Prokaryotic cells are going to be cells that don't possess a nucleus, so they're lacking a nucleus. This is going to be pretty much bacteria. Eukaryotic cells are what we are made up of, and so these are much larger cells, and cells that actually have a nucleus in them and a lot of organelles. And so eukaryotic cells do start off, some are single cellular, such as amoebas, and certain protozoa you can find are eukaryotic cells. But such as ourselves, we're multicellular organisms that are made up of eukaryotic cells. So we're made up of trillions of these eukaryotic cells. And so the main thing here is that there's actually a distinct nucleus when you start looking at eukaryotic. Cell theory was originally put together by a man named Robert Hooke. He lived in the 1600s, and he made a microscope, and he was actually looking at things for alchemy. Back then they actually believed you could actually change ordinary things into gold, and so a lot of your early microscopes were actually built in trying to study what substances were made of, trying to turn things like rocks into gold. So he got bored one day and decided that he would take a thin slice of some cork, and he put it on the slide, and he looked at it, and he noticed that inside of this cork there were these little bitty square areas in there that he could see, which we now know were cells. And what they looked like to him was little bitty rooms, and so kind of like a dungeon, and so or a jail. And so that's where the word cell came from. Schlenden and Schwann, somewhere in the early 1800s, started a theory that said that all living things are composed of cells. Actually, it was a hypothesis that later turned into a theory. One thing to know that in biology, all of our laws are theories unless they have to deal with chemistry or physics. Physics have hard, firm laws based in mathematics, so does chemistry. So if we're looking at something like Wolff's laws in biology, Wolff's laws has to do with gravity and the effect on the body. And so it's based in physics off of a man named Wolff made as far as laws in physics, and we've brought those over to biology. Therefore, we still use the word laws. Same thing when we look at Starling's laws, which he was a chemist. We actually will use some of his laws of chemistry, once again, that are based in mathematics in biology. But when we come up with something just in biology, then that ends up being, as if it's a law, it's called a theory. And the difference between the two of why we call them theories and not laws is that every organism on planet is slightly different. And so something like in chemistry, you can have, if you remember in acid-base balance, we were doing a video from Khan Academy, and he had Avogadro's number. And so Avogadro's number is a law, and it's based on a mole is a certain number multiplied by 6.023, and so that's a constant that never changes. The thing with biology is it always changes. So therefore, when we look strictly at biology, it's hard to have a very hard set firm law in place because every organism is different. You don't have a mathematical constant that you can actually keep right there. So therefore, we end up using the word theory to mean the same thing as laws. And of course, if it's something that we're hypothesizing, that's totally different than what a theory is. So when we talk about cell theory, this is actually a law. And so Schlinden and Schwann actually sat there and developed this hypothesis that later on became the cell theory, that all living organisms, be they plant or be they animal, are made up of, or fungus, are made up of cells. Virchow took that a little bit further, and he lived somewhere towards the late 1800s, early 1900s. Or no, I take that back. He lived somewhere in the mid 1800s, and he actually expanded on this and actually said that cells actually divide and arise from pre-existing cells. And so that was a very revolutionary thought at the time that Virchow came up with. There is a, right here on the slide, this is wrong. That should be the symbol for a micron. So animal cells are usually 10 to 30 micrometers, not millimeters, but micrometers in diameter. So a micrometer is a thousandth of a millimeter. And if you want to know how wide a millimeter is, get a dime, and a millimeter is the exact width of a dime, or I should say a dime is the exact width of 1 millimeter. So a micrometer is a thousandth of the width of a dime. So these things are very small. You can't see cells actually visibly. And the reason for this is because of surface area. There's actually a relationship here between surface area and the volume that can be within a cell, and it has to deal with the fact that cells need to absorb as much nutrients as they can, such as oxygen, glucose, amino acids, and all those things. So they need surface area to actually bring those across. But to actually penetrate into the center of the cell, if they're too large, the nutrients can't actually diffuse into where they need to be at. So that's the reason why they keep such a smaller size. And by having a smaller size, they can still do certain structural features that actually increase their surface area and their absorption rate. At the same time, having the smaller size also makes it easier to get rid of waste products such as lactic acid and carbon dioxide. So small cells actually do have a proportionately larger surface area which they can absorb things through compared to a larger cell, and it has a large surface area too, but proportionally speaking, smaller cells actually can have almost as much surface area as a larger cell will. There are some cells that are going to be larger in the body, such as muscle fibers or nerves. They can be very long, but the diameter of them usually does not vary much from the 10 to 30 micrometers that we're talking about. So some of these larger cells that we're looking at, like muscles, can actually be multinucleated because a single nucleus cannot actually control the metabolic rate. So they have to have multiple nucleuses. So when we actually start looking inside the cells, there's some anatomy to know about on the inside and the outside. And so probably the first thing to talk about is actually the cell membrane, which is the outer structure of the cell. And so this cell membrane is actually what's going to surround and protect the inner workings of the cell from the external environment. You can almost think of it as like the walls of your house. They actually keep your air conditioning on in the inside to a comfortable level for you, keeps everything in balance, or the heat in the wintertime for you. And then there's certain places that you can enter and exit your house from, and you can open and close windows and things like this. Cell membrane kind of does that. It's not rigid like the house. It actually moves around. Things with cell walls are very rigid like plants and fungus, but mammals and all the animals are made up, insects, fish, we're actually made up of cell membranes. There's no cell walls, so that gives us flexibility, but the membrane's there to actually keep homeostasis in place. and it's actually to protect what's going on the inside from interference from the external environment. You have the cytoplasm, which is going to be made up of water, electrolytes, and then different organelles. And then finally, we count differently as the nucleus. And the nucleus is going to have its own membrane around it. That's going to be like the cell membrane as far as its composition is made up of, or very similar to it. And so that's going to house the DNA in there, and it's actually going to protect the DNA from things going on in the cytoplasm that could be harmful to it. Everything inside the cell membrane other than the nucleus is known generically as cytoplasm. So that's a lot of these structures we're going to be talking about all together are going to be known as cytoplasm. And so there's the proteins, the electrolytes, cytoskeleton, organelles. All these different things are going to be found inside the mammalian cell. Cell membrane is going to be made up of a lipid bilayer, and it's going to actually have different proteins and lipids and cholesterols embedded in there as rafts. And it's very flexible. It's an elastic barrier, and it keeps what's inside the cell and the cytoplasm in, keeps it protected from the outside environment, and then it keeps the things on the outside out. And so it's a semi-permeable membrane. It's going to let certain things pass through at certain times and keep certain other things out. So it governs movements of atoms and molecules in and out of the cell. And so the primary thing it's made up of is really, it should be phospholipids, should be in front of proteins right here, because the main thing it's made up of is phospholipids. And then you have proteins, cholesterols, you have glycoproteins, you have lipoproteins, you have lipids, you have carbohydrates. All these things are put together inside the cell membrane. So this lipid bilayer is very important, and so we'll look at a slide in a second here, and we'll actually look at the hydrophilic heads that have the phosphate, that have a slight negative charge, so they attract water, and the fatty acid tails, which are neutral, which are hydrophobic and actually repel water. And this whole thing is fluid mosaic. We'll see some videos, and you'll actually see where this thing's constantly moving, and so it's actually made to where these lipoprotein molecules are constantly moving around. So are some of your proteins, your lipids, and so things are actually meant to where they can actually move easily around the cell to where they can actually do their certain functions that they need to do as far as physiologically. Some lipids will pass through easily. One thing they do have to get by is these little charged phosphate heads right here, which do repel lipids, but if they can actually get past it, then these neutral fatty acid chains that come off of the phosphorus head here on a phospholipid, then they can actually pass easily through. These are little molecules of cholesterol, and so usually it's at these points where you can actually get lipids to pass through. We'll actually be talking about some of the things like this is a peripheral protein. Some of these can be all the way through. Some of these can just be partially through. And an integral protein. And the main difference between a peripheral protein and an integral protein is integral proteins will always have a channel that passes through them. And a peripheral protein can be structural or globular, and it doesn't have a channel that goes all the way through. A lot of these actually will have things like this is actually a chain right here of carbohydrates, and so they'll actually be like a glycoprotein. By having the fatty acid chains right here, it actually makes it to where water doesn't pass through as easily. Usually it has to go through the channels, and that actually kind of keeps the cell's moisture level steady and keeps it from dehydrating, and so that's a very important thing. When we look at the membranes, we were talking about the different proteins here. So you have the integral proteins. They'll span the entire width of the membrane, the cell membrane right here with the phospholipids, and they have a channel going through them. What you actually are seeing here is a peripheral protein. Some of these can be attached to the cytoskeleton, especially if they're needing to stay in one place. Some of these can be bound to just the outer portion of the lipid bilayer. Others can be bound to just the inner portion of the lipid bilayer. And a lot of times these are looking for chemical signals to make them do something. If they're bound to just this inner layer here and they don't go all the way across as a peripheral protein, maybe there's a chemical signal that will actually make it go across and actually release that protein outside of the cell. On the other hand, some of them that are actually just bound to the outer part of the layer, if a hormone comes along, it can actually trigger a change in that protein, and that protein can actually then pass all the way through the lipid bilayer and actually enter inside the cell and actually give a chemical messenger from that hormone. So there's a lot of different ways that these peripheral proteins can be. They can be fixed, they can be moving, they can do multiple functions. One thing that we look at is what they call a glycocalyx, and so this is actually a protein with a sugar on it, and this is to hold two cells together. So this is a cell and this is a cell right here. And so this protein right here has actually got a sugar coming off of it that they call a glycocalyx, and so this sugar right here attaches to this protein on the other side, and this is how the cells are actually bound together, and so it actually increases adhesions. And then we also have things like other carbohydrate molecules right here, or potentially cholesterol molecules too, that are actually trying to adhere these two cells together. A lot of times you also have this glycocalyx can also be used as membrane receptors. So kind of like out here on this one, a lot of times these can actually be looking for specific hormones, and that can cause changes in adherence of that molecule or that drug even to the cell. And so that's a very important thing, is these glycoproteins to do that. Something as far as propulsion and movement with cells that we look at is something called cilia and flagella. So all of these are going to be made up on the outside of the cell membrane. So you're going to have the actual cell membrane out here. And then the inside, you're going to have 9 pairs of little microtubules in here. And they're actually going to cause movement. Now, the difference between these cilia and flagella is the number. And when we look at things like cilia, there can be hundreds of these on the surface of a cell. And they all will move in one direction. So where we find a lot of this is going to be like the reproductive tract, the respiratory tract, the GI tract. So in the respiratory tract, what these actually do is they actually collect mucus that's produced, and that mucus is going to collect things like dust, bacteria, allergens like pollens. And so that mucus is going to trap all that, and then the cilia is actually going to move that mucus up to the back of your throat to where it can be swallowed or coughed out. And they always move in one certain direction. And the little microtubules in here that are going to attach down in there, they're actually going to attach to the center of the cell in something known as the centrioles. Flagella, on the other hand, are singular, and they're used for propulsion of the cell. And so the only flagellated cell that we see in mammals and avians and most animals is going to be sperm. And so spermatozoa has a single flagella off of them. So cilia, there's multiple, there's hundreds of these, but flagella, there's only one. When we look at cilia, cilia is to move things like mucus or something along like a conveyor belt. Flagella are here to strictly move the cell. They're to transport the cell from one location to another. The cytoskeleton, this is actually a framework for the cell, and so this actually is what gives the shell structure and shape. And it can also serve many other functions such as transport. So we'll actually watch a video where we'll look at these little motor protein molecules that will actually move substances from one part of the cell to the other. They actually help to anchor organelles, and they actually provide direction for certain metabolic activity. And so I show 3 different fibers, but there can actually be 4 different fibers involved when we actually start looking at the cytoskeleton. And so the largest one is going to be the microtubule. This is 25 nanometers. So a nanometer is actually a millionth of a millimeter. So these things are small, but the largest of them is this microtubule. And so this microtubule right here is made up of tubulin subunits, molecules that are put together, and they actually have a certain charge to them, and so they easily go together, and if you undo the charge, then these things actually break apart fairly easily too. And so these are kind of comparable to like the big pole in the middle of a tent. If you're putting up a tent and you have a large pole, these provide the main framework structure, and they're constantly being changed around as the cell's needing to change shape. But they provide the main structure, and everything else is going to kind of grasp onto these microtubules. Next, you have these intermediate filaments. These are usually made up of the microfilaments up above, and they're woven together very tight. And they're about 10 nanometers, so they're about half the size of a microtubule. And these actually kind of make a framework off of the microtubule and go out to other areas. So they're kind of like the string that goes between those poles that kind of helps prop it up. So you can almost think of them as a guide wire. And then finally, coming off of these intermediate filaments and the microtubules too, you have these microfilaments, which are just smaller ropes, and they're about 7 nanometers inside. One thing is the intermediate filaments are probably much tougher than these microfilaments are because of the way that they're actually woven together and spun, and so they give a lot more tensile strength. One of the things, and I don't have it on here, is something called a microtrabecula. In the book it says that some scientists think it exists and it makes a fine meshwork, and we've pretty much figured out that they do exist, and they're probably about 2 to 3 nanometers inside, so they're half the size of the microfilament. But all these together make up the cytoskeleton. When we look at organelles are going to be inside the cell, and they're going to actually be specialized little micro organs in there that have a specialized purpose. So they're going to do something specifically to help maintain homeostasis within that cell. And they're actually going to have a membrane that's actually going to surround them, and it's going to be a lipid bilayer membrane, just like the cell membrane that we looked at, that's actually going to enclose and define the structure. And these are going to be located throughout the cytoplasm. So the first one that we get to is mitochondria. And so mitochondria is known as the powerhouse of the cell. So this actually makes the ATP that the cell needs in order to do its function. So it makes energy for the cell. What's really unique about mitochondria is they actually have their own DNA within inside them. And they actually are about the size of a bacteria, and they can actually move freely around on the inside of the cell. And so they can actually go to an area of the cell that's needing more energy produced. Aerobic metabolism happens here, and this is the huge leap forward that actually makes it possible for us to live, because they increase energy levels for the cell by greater than 95%. The outer coating of them is smooth, and inside is very convoluted. And so we have the outer membrane out here, and then we have what we call the cristae on the inside. And actually this cristae is convoluted because it's needed to be this way for surface area to actually make the ATP and to do aerobic respiration to make energy for the cell. By it having its own DNA, is very important, especially when we're looking at diseases into the future, because mitochondria is passed down from only the mother, not from the father, but from the mother to their offspring. And so we're finding that mitochondrial DNA and mitochondria actually do play roles in diseases such as cancer. And we're looking further and further into how much of a role this actually plays. And so things like certain treatments for mitochondria might actually in some ways cure some cancers in the future. Ribosomes, these are small little dots, little subunits, and these actually have RNA in them. And there's a larger unit on the outside. And what they do is they actually have what we call ribosomal RNA. This is going to attach to what we know as messenger RNA. And it's actually going to read that messenger RNA. And when it reads it, it's actually going to grab each time that it reads a little codon or a little 3-section segment of that RNA, messenger RNA. It's going to grab an amino acid that it codes for, and it's going to actually start putting these amino acids together and eventually start making proteins. And those proteins are going to actually direct cell metabolism. Ribosomes make proteins intended for use inside the cell, and these are usually freely distributed throughout the cytoskeleton. And then also proteins made for the plasma membrane are meant for export, and these are usually going to be into something that we know as the endoplasmic reticulum, and specifically what we call the rough ER, the rough endoplasmic reticulum. So when we look at ER, there's two types of it. You have rough and smooth. And so this is endoplasmic reticulum. This up here is a Golgi apparatus, and we'll get into what the Golgi apparatus does in a few minutes. But you see all these little blue dots. These actually are supposed to be ribosomes, and they're attached to what we call the rough ER because they have a rougher appearance. So if you have a ribosome floating out here, it's making proteins for the cell, but when they attach to the endoplasmic reticulum, these are going to be usually exported or moved up here to the cell membrane. And so rough ER is just a place where these ribosomes can attach to, make these proteins, and these proteins are actually going to be transported and stored through here, packaged, and then they're going to be moved on up to the Golgi apparatus. And then when we actually sit here and look at the smooth ER, so we have the rough with those little ribosomes, the smooth ER stores nutrients for the cells, makes lipids, and it can also be a place of detoxification. So if you have any toxins or anything that the cell wants to get rid of, it will bring it to the smooth ER, and the smooth ER will actually put an envelope around it, send it to the Golgi apparatus, and the Golgi apparatus will actually get rid of it out, send it up to the cell membrane to where it can be excreted from the body or from the cell. And so the smooth ER plays a very important role as far as detoxification. But like I said, it also does storage, keeps lipids, keeps a lot of essential nutrients actually stored on the inside of the cell. Golgi apparatus, its whole job is to sit there and take things from the ER, smooth or rough, and then it's going to repackage it, and it's going to package it to where it can actually be taken and either used in the cell as a vesicle to store things, or in this case to be a lysosome. So lysosome would actually be used something to dissolve something like this. Could be bacteria, this could be a white blood cell, and so if this bacteria is brought in, this lysosome package is going to come over to that bacteria that's been engulfed by that white blood cell, and then those lysosomes are going to be released and they're going to kill that bacteria. So it packages things like lysosomes. It actually packages things also for export out of the cell. And so its whole thing is it actually modifies and packages and actually then sends, usually through those motor proteins I was talking about, things to be placed either in the cytoplasm like the lysosomes to work, or it's going to be taken to the cell surface to where it can be excreted out of the cell. So lysosomes, these are going to be specialized vesicles. So there's a lot of different vesicles in the cells. They can be used for storage of things like lipids and fats through the Golgi apparatus. They can be made for waste, but we have peroxisomes and lysosomes, and a lot of these times these things will actually be used to either dissolve substances inside the cell or on white blood cells to actually take care of foreign invaders. And the lysosome can be known as the suicide sac, because if all these things were to actually rupture at once, it will actually kill the cell. Most of the time these things are kept fairly well under control to where they're only released when they're needed to break something down. But if they all happen to go at the same time, the enzymes and the proteins actually put in here would actually destroy and kill the cells. Peroxisomes, they actually have a little bit of peroxide in them. And they're used to actually remove things called free radicals, and they can be used by white blood cells to actually kill things such as anaerobic bacteria. And so they actually catalyze things with hydrogen peroxide and convert it to water. And so when you convert hydrogen peroxide to water while they work on anaerobic bacteria, you actually release oxygen, and that oxygen kills anaerobic bacteria. Inclusions are substances in the cells that the cells actually brought in, and so these can be vesicles, vacuoles, granules, different things like that that are membrane bound. In the case of secretory granules, vacuoles, and vesicles, these things can actually be made at the Golgi apparatus and then stored in the cell for use at a later time. Centrioles, these are small hollow cylinders within the cell, and their main job is to be an anchoring point for the cytoskeleton. They're very important in division of the cell, and when we get into things like mitosis and meiosis, we'll find out why they're very important. And you can actually see these during division of the cell, and we'll see that. But these are meant for the cytoskeleton to actually attach itself to. The nucleus is going to be in the cell, and it's a dark staining part of the cell, and it usually stains purple when we look at cells. And so they can be spherical in shape, they can be oblong, they can have different shapes, they can be singular, segmented, multi-segmented. They have a lot of different shapes. And there can be in like muscle cells, which can be quite long. They can actually have multiple nucleuses inside of those cells. And so their function is they carry the DNA, and the DNA for actual function of the cell, so maintaining homeostasis by actually coding for proteins that are needed to control cellular activity in the cells. And the second thing they do is they actually carry that hereditary information, that genetic information, to pass on to the next cells that are actually going to divide or arise from them, or onto offspring, such as when we get into things like meiosis and we start looking at gametes or sex cells, being able to send that genetic material on to the next generation so that the organism can survive and the species will continue to go on. One thing to know is that we do have some cells that are actually lacking a nucleus, and the main ones that we see are mature mammalian red blood cells. So you can know the difference between a dog, cat blood cell from those of a snake or a chicken, because snake and chicken blood both have nucleated red blood cells, whereas all mammalian blood is anucleated. So actually when they mature, they actually lose the nucleus. And this is to actually make the cell smaller and actually pass through smaller capillaries and be more flexible by lacking the nucleus. And so it actually gives them a longer lifespan and gives it a greater oxygen carrying capacity, and actually makes it to get to where it can get into a lot more tighter areas and a lot more, because the cell's a lot more flexible without the nucleus. When we look at the nucleus, we're going to have the outer envelope or membrane, and so this is going to be made up of a lipid bilayer. You're not going to really have a lot of the different types of proteins except for channel proteins right there, or integral proteins in the surface to make pores. To allow things such as RNA to pass out, certain chemical signals to come in. But it pretty much is there to protect the cell nuclear envelope or cell nuclear membrane, is there to protect the DNA from things in the cytoplasm, especially some of the different toxins that can actually build up, things like lactic acid that could potentially damage the DNA. We have nucleoplasm, which is a gel surface substance made up of water, electrolytes, and then the different DNAs and RNAs and stuff. It's very similar to cytoplasm. You just don't have the different organelles and some of the larger protein molecules and things like lysosomes and stuff in the nucleoplasm that you'd find in the cytoplasm. You have something called the chromatin, which is actually the genetic material, the DNA, and then you have an area called the nucleolus, or you'll hear it called the nucleolus. So this is a very active area of the DNA where you're actually making things like ribosomes and RNA, and it actually has a darker staining to it than the rest of the nucleus does when you look at it with the stains. So you have these little nuclear pores in here. They're usually made up of protein, channel proteins, and it does allow things like RNA to pass in and out, certain proteins to pass in and out. But it's there to keep out some of your larger proteins like your lysosomes and things that could be detrimental to the DNA. A lot of times some of these pores will actually be continuous with the endoplasmic reticulum, especially the rough ER, so that some of the RNA will actually pass into there to actually be read by those ribosomes that attach to it and to help to make those proteins. And a lot of times your ribosomal RNA is actually made in that area of the nucleolus, and so that nucleolus, that ribosomal RNA can actually pass out through here and actually attach to the other subunit to help make up ribosomes. So RNA is actually able to freely pass in and out. So does your messenger RNA. It passes freely in and out through these pores. DNA and RNA, of course, made up of nucleotides. We talked about that in chemistry. So they're going to have a nitrogenous base, a pentose sugar, and a phosphate group. And so there are things known as cytosine, adenine, uracil, and they're going to be put into a chain. And the DNA is going to be a double helix, and it is the master code. It keeps all the information genetically needed for the cell. And then RNA is going to be made, those nucleotides, to actually send a signal from the DNA out to those ribosomes to have certain proteins made. So remember that DNA here is a double helix in shape. RNA is usually more of a single helix in shape. And DNA is going to be the master code, and then RNA is going to be read off of this. So this helix would actually split off, and you'll actually have, we'll get into this, of how transcription occurs, where you'll actually sit there and it'll actually read sequences of the DNA and actually make RNA to match it. And then that RNA is actually going to go out. and be read by the ribosomes, and a protein will be made. So chromatin is that light and dark staining area of the nucleus made up of DNA and what they call histones. Histones play an important role in gene regulation. A single strand of DNA winds 8 histone molecules, forming a granule called a nucleosome. And the nucleosomes are held together by short strands of DNA called linker DNA. So this is actually what wraps it up so tight and makes it to where when you look at it underneath the microscope and you see a nucleus stain, that it has this dark stain and very bound together very tightly. The nucleolus is that dark staining patches within the DNA, not membrane bounded whatsoever, but RNA subunits are being made here, ribosomal subunits are being made, and so it's a very active part of the DNA. There's a lot of transcription of the DNA going on, and a lot of things that are actually being formed in that area as far as RNA goes and ribosomes go.